Prentice Hall, in conjunction with Blossom Medical Communications, has produced this video tutor to complement the Fundamentals of Anatomy and Physiology textbook by Frederick Martini, although you'll find it very useful no matter what text you're studying. The concepts you'll learn in the next 75 minutes will also apply to other related courses, like anatomy, physiology, and introductory biology. Sometimes visualizing physiological processes can be difficult. The text provides a solid foundation, but couple that with a detailed 3D animation and you're off to a good start. Video footage is implemented to build analogies to further explain each concept. At the beginning of each segment is a list of concepts you should understand before viewing the segment. Also, each segment gives an overview of the major topics to be presented. In order to pace yourself, quizzes called concept checks are added after each section. If you are unable to answer the questions, it may mean that you should rewind this section and view it again. We hope that you'll find the video tutor as an invaluable study tool. Welcome to Prentice Hall's video tutor section on protein synthesis. By now, you know that the nucleus is an information storage center and that DNA holds that information. In order to understand protein synthesis, you might think of the nucleus as the headquarters of a manufacturing complex. The information needed to produce all of the protein products of the cells is located in the nucleus, stored in the form of DNA. Outside the nucleus, in the cytoplasm, are the raw materials, the amino acids, as well as the metabolic machinery that actually builds the proteins by stringing amino acids together. DNA must remain inside the nucleus. The two questions raised then are, how is the information in DNA transferred to the cytoplasm? And how is that information then used as a blueprint for polypeptide synthesis? To begin to answer some of these questions, we will look at the molecules and processes involved in protein synthesis. First, we'll describe the information storage system of a cell, the cell's DNA. Then we'll look at transcription, the process by which the information is copied to RNA so that it can travel to the cytoplasm. Finally, we'll describe translation, where the information in RNA is used to assemble amino acids into polypeptides and proteins. Each person's hereditary information is encoded by the DNA inside their cells. Half of the DNA was inherited from the mother, and half was inherited from the father. The DNA is stored in cell nuclei. From its position in the nucleus, DNA controls cellular anatomy and physiology. This in turn determines the structure and function of the body and influences individual development and behavior. Thus, by controlling protein synthesis, DNA determines how an organism is built and how it functions. A DNA molecule consists of paired DNA strands that twist around one another to form a ladder-like structure called a double helix. Each strand is composed of a long chain of nucleotides, each of which contains a nitrogen base. Bases from one strand form hydrogen bonds with the bases in the opposite strand, forming the steps of the DNA ladder. The two DNA strands are complementary. One DNA strand, the coding strand, contains the information that specifies amino acid sequence in a polypeptide. The other DNA strand, called the template strand, is used to make the blueprint from which the polypeptide eventually will be made. There are 4 different types of nitrogenous bases in DNA: guanine, cytosine, adenine, and thymine. In RNA, thymine is replaced by a base called uracil. Because guanine can only pair with cytosine and vice versa, these two bases are said to be complementary. Likewise, thymine and adenine are complementary base pairs. DNA's information is stored in the linear sequence of its nucleotide. The fact that each nucleotide can pair with only one other type of nucleotide is important. Complementary base pairing forms the basis by which a DNA strand can be used to create an RNA molecule that retains the information of that linear sequence. Let's take a moment now for a concept check.
How does DNA store information? What feature of DNA enables its information to be copied? Which DNA strand specifies the amino acid sequence in a polypeptide?
Welcome back. Now that we understand how information is stored in DNA, let's explore how this information is used to make proteins. All the nucleated cells in the body, except for sperm and eggs, have exactly the same DNA. But some cells make hair, while others make eye color. This is because the different cells are using different genes to guide protein synthesis. A gene is a segment of DNA that contains all the information required to make a polypeptide. Each gene contains a specific sequence of nucleotides that determines the order in which amino acids will be linked together to form the polypeptide. As you recall, polypeptides are manufactured in the cytoplasm. Transferring the information in a gene to the site of protein synthesis requires RNA. There are 3 types of RNA in the cell. In order to understand the way in which these 3 types of RNA interact, let's return to our analogy of the cell as a manufacturing plant. To produce your product, you must create a blueprint, gather the raw materials needed, and have the machinery needed to assemble the parts. Each type of RNA does one of these 3 separate jobs. Messenger RNA, or mRNA, is the blueprint for polypeptide synthesis. Along the mRNA strand, each group of 3 consecutive nucleotides, called a codon, specifies a single amino acid. The sequence of codons along the length of the mRNA strand determines the order of amino acids in the new protein. Transfer RNA, or tRNA, brings in the raw materials for protein synthesis by binding to specific amino acids in the cytoplasm and carrying them to the ribosome for the assembly of amino acids into protein. Ribosomes, the protein factories of the cell, are made of 1 light and 1 heavy subunit. Each subunit is made of proteins and ribosomal RNA, or rRNA. The events leading to protein synthesis can be divided into 2 stages, transcription and translation. In transcription, the template strand of DNA is used to make a complementary strand of messenger RNA, which then carries the gene's instructions to the cytoplasm. During translation, ribosomes use the information in the messenger RNA to assemble proteins. Although we will focus here on mRNA when we discuss transcription, you should remember that any RNA, whether it is messenger RNA, transfer RNA, or ribosomal RNA, must be synthesized through transcription of a DNA template. Now let's see how all these nucleic acids interact during transcription and translation. In order to transcribe DNA, the 2 strands of the double helix must first be separated to expose the template strand. Once this occurs, nucleotides already present in the nucleus begin to pair with the exposed bases with the help of an essential enzyme called RNA polymerase. This binding occurs only between complementary bases. Guanine in the DNA template will pair with cytosine in the growing RNA strand. thymine will pair with adenine, and so on. Remember, however, that RNA incorporates uracil rather than thymine, so for every adenine in the DNA template, a uracil will be added to the RNA. RNA polymerase continues to link nucleotides until the mRNA is complete, after which the mRNA separates from the DNA template and moves out of the nucleus. Once in the cytoplasm, mRNA carries its information to a ribosome. Keep in mind that the ribosomal subunits exist separately in the cytoplasm. Before assembly of a polypeptide can begin, the ribosomal subunits must combine around an mRNA strand. This is a good spot for a concept check. Take a few moments now to answer 4 concept check questions.
What are the 2 major processes of protein synthesis? What are the 3 types of RNA? Which type of RNA carries the blueprint for protein synthesis? What is the function of tRNA?
Welcome back. Now that we have learned about transcription, the first major process in protein synthesis, let's move on to see how translation enables our cells to make proteins. Every day our bodies carry out varied tasks such as digesting food, growing fingernails, and making skin cells. These tasks and numerous others require proteins. As you know, the polypeptides that make up a protein are made in the process of translation. Translation occurs on ribosomes and requires the coordinated efforts of messenger RNA, ribosomal RNA, and transfer RNA, as well as numerous proteins. The first step in translation, initiation, begins when an mRNA binds to a free light ribosomal subunit. A transfer RNA molecule then brings the first amino acid to the light subunit of the ribosome. Other protein factors join this assemblage, and then the heavy ribosomal subunit combines to complete initiation. Transfer RNAs continue to bring in amino acids in the order specified by the sequence of nucleotide bases in the mRNA. For each possible triplet combination, there is a specific tRNA that will bind to it. Amino acids floating in the cytoplasm are picked up by tRNA molecules and delivered to the ribosomes. An amino acid attaches to one end of the transfer RNA molecule, while on the other end are 3 exposed bases. Because these bases attach in complementary fashion to the 3 bases in an mRNA codon, this region of the tRNA molecule is called the anticodon. Only a tRNA with a specific anticodon can pick up a specific amino acid. Many types of tRNA molecules exist, and each has a different anticodon to carry one of the 20 different amino acids needed for protein construction. Thus, the sequence of codons in the mRNA strand by binding only complementary tRNA intercodons determines the sequence of amino acids in the newly synthesized polypeptide. In the initiation step, as the first tRNA approaches the ribosome, its anticodon binds to the mRNA codon at the first of 2 binding sites on the light ribosomal subunit. Elongation is the second step in translation. After initiation, a second tRNA attaches to the second mRNA codon, which occupies the other binding site on the light subunit. An enzyme associated with the heavy ribosomal subunit catalyzes the removal of the amino acid from the original tRNA and attaches it to the amino acid on the newly arrived tRNA. In this process, a peptide bond forms between amino acids, creating a dipeptide. The original tRNA detaches from the mRNA and leaves the ribosome, which shifts or translocates 3 bases to the right along the mRNA strand. This brings a new codon to the binding site on the light ribosomal subunit. When a tRNA with the appropriate anticodon binds to this codon, it will deliver the next amino acid to be added to the growing peptide chain. This cycle repeats until polypeptide formation is complete. In the last step of translation, called termination, a stop codon signals termination factors to help cut the polypeptide off the last tRNA and release it from the ribosome. Once made, a polypeptide chain, singly or in combination with other polypeptide chains, assembles in the cytoplasm into a new protein. Let's pause here for a final concept check.
By what process is a polypeptide made in the cytoplasm of a cell? What specifies the sequence of amino acids in a growing polypeptide? What is the final step in translation? What is translocation?
I hope this Prentice Hall video tutor section has helped you understand the components of protein synthesis. Welcome to the Prentice Hall Video Tutors segment on membrane structure and function. In this section, we explore how the cell's membrane controls what goes in or out of the cell. Cells are the structural and functional units of living organisms, whether tall and thin or wide and flat, whether transporting oxygen or absorbing nutrients. Cells share common structures. One of these is the cell membrane. To begin to understand how cell membranes function, we will first examine some basics of membrane structure. We will then discuss diffusion and osmosis and how these processes move substances across membranes. Finally, we'll take a closer look at carrier proteins, the proteins that exchange molecules across the cell membrane. Like the walls of a house or building separate the interior from the outside world, the cell membrane is a barrier that separates the contents of the cell from the external environment. Cell membranes are a mosaic of protein and lipid molecules, both of which can drift from place to place within the membrane. Most of the surface area of the membrane consists of a type of lipid called phospholipid. Each phospholipid molecule has a polar head and a nonpolar tail. The phosphate-containing head is hydrophilic or water-loving because its polar structure is attracted to water molecules, which are also polar. The fatty acid tail is hydrophobic because it is nonpolar and repels water molecules. Cell membranes are phospholipid bilayers, a double-layered structure in which the hydrophilic heads of the phospholipids face outward. The middle of the membrane contains the hydrophobic tails of the phospholipids, an arrangement that shields them from contact with water molecules. Whereas membrane lipids act as a general barrier around the cell, membrane proteins have specific functions. For example, they may act as enzymes, receptors, anchors, or channels. There are 2 major populations of membrane proteins: integral proteins, which are part of the membrane structure and usually span the width of both phospholipid layers, and peripheral proteins, which are bound to the surfaces of the membrane. Understanding the basic parts of the cell membrane is important. Before we move on, let's take a moment to answer 4 concept check questions.
What separates the interior of a cell from the external environment? What are the major components of a cell membrane? Phospholipid molecules have a polar and nonpolar end. How does this affect how they interact with water? What are some functions of membrane proteins?
Now that we know what cell membranes are made of, let's look at how they work. Like bumper cars, molecules are in constant motion, colliding, then bouncing off each other. When bumper cars collide, they scatter into less crowded areas. They spread out from an area where they are highly concentrated to an area where they are less concentrated. Diffusion is the process by which particles in a substance move from an area of relatively high concentration to an area of relatively low concentration. Although molecules move freely in a solution, they may not be able to pass through the cell membrane, which is permeable to some molecules but not others. This semi-permeability of a membrane is important to its function. But before we go further, let's continue our discussion of diffusion by defining some key terms. Solutes are particles that are dissolved in fluid. If the particles are not distributed evenly throughout the solution, the difference in the solute concentration between two areas in the solution is called the concentration gradient. The random movement of particles by diffusion in a solution always causes them to move from an area of higher concentration to one of lower concentration, or down their concentration gradient. As this movement continues, the particles eventually become distributed evenly. In other words, diffusion takes place until the concentration gradient is eliminated. At this point, where there is no net movement of particles in any direction, equilibrium has been reached. Random molecular motion still continues, but the overall distribution of solute remains even and constant. Dropping an ink drop into a container of water demonstrates diffusion. Ink molecules are highly concentrated in a drop, but not elsewhere in the container. The ink molecules in the container behave like bumper cars, randomly colliding with each other and with water molecules. The ink molecules diffuse from the drop where concentration was highest to areas of lower concentration. The ink molecules diffuse until equilibrium is reached and the ink is distributed evenly throughout the container. Here is a schematic look at diffusion of an ink drop. There is net movement of ink molecules from an area of high concentration to one of low concentration. One important point to remember is that diffusion is a transport bargain for cells, since diffusion occurs without any energy input by the cell. An essential feature of cell membranes is that they are selectively permeable, meaning some materials pass through them freely, while other materials cannot. Passage may be restricted by a molecule's size, shape, and/or electrical charge. For example, fat-soluble molecules such as steroids, alcohols, and some vitamins can dissolve in the lipid portion of the membrane, which is how they diffuse into and out of the cell. Water-soluble molecules, however, cannot pass through the hydrophobic middle portion of the phospholipid bilayer. Instead, they must diffuse through the membrane channels. Even then, only ions or very small molecules can fit through such channels. The channels formed by membrane proteins are like doorways through which molecules enter and leave cells. Membrane channels, like doorways, come in several types. Some doorways have no doors, so they are always open. Membrane channels that are always open are called leak channels. Some doors open and close. Channels that open and close are called gated channels. Other doors allow movement in and out at the same time, exchanging someone going in for someone going out. Membrane proteins that do this are called carrier proteins. Only solutes of a particular type can pass through a given channel. As with all diffusion, when molecules flow down their concentration gradients freely across the membrane, the cell does not have to expend energy. Take a moment now to answer a few concept check questions.
What is meant by selectively permeable? What are some determinants of whether a molecule can cross a phospholipid bilayer? How much cellular energy is required for a cell to gain or lose substances through diffusion? What macromolecules of a membrane form channels?
Welcome back. Now that we've looked at diffusion of solutes, let's take the time to look at diffusion of water across a membrane, a process called osmosis. Like other molecules, water molecules move from an area of high concentration to an area of lower concentration. Osmosis occurs when water diffuses across a membrane that is freely permeable to water but not to solutes. In this container, two solutions are separated by a membrane permeable only to water. The concentration of the large yellow solute molecules is higher in solution B than it is in solution A because there are fewer solute molecules in solution A. The inverse is true of the water. The concentration of water molecules, represented by small blue spheres, is higher in A than in B. The way in which water will behave in this example is similar to how osmosis occurs in cells. The solute molecules are too large to pass through the pores of the membrane. However, water molecules freely move across the membrane. Water molecules move down their concentration gradient, moving across the membrane from the side with the higher water concentration to the side with the lower water concentration. This increases the volume of B. Water movement continues until the solute concentration on both sides of the membrane is equal. For our discussion, we are ignoring the counteracting effect of the weight of the column of water. Osmosis across the cell membrane depends on the tonicity of the cell's environment. Tonicity is the term used to describe what effect a given solution will have on the tension of a cell membrane placed in that solution. Tonicity depends on the difference between the solute concentration on the inside of the cell versus the outside. Fluid that normally surrounds cells is isotonic. Iso means equal, and when the concentration of solutes is equal on both sides of the cell membrane, no net movement of water occurs into or out of the cell. Hypo means below, and a hypotonic solution is one that has a lower solute concentration than the cell. Consequently, a solution that is hypotonic will force water across the cell membrane into the cell. The cell swells and eventually may burst or lyse. Hyper means above, and a hypertonic solution is one that has a higher solute concentration than the cell. Consequently, an extracellular solution that is hypertonic will force water to move out of the cell, causing the cell to shrivel or crenate. Are you ready for another concept check? Take a moment to answer the following three questions.
What is osmosis? What is an isotonic solution? What is a hypertonic solution?
Welcome back. Let's now take a look at how some other molecules cross the cell membrane. Most molecules needed by our cells are not lipid soluble, and they are too large to pass through membrane channels. Instead, carrier proteins pick up molecules on one side of the membrane and release them on the other side. Each carrier protein transports only specific molecules or ions across the membrane. In facilitated diffusion, movement is down the concentration gradient and requires no energy expenditure by the cell. Most sugars, as well as all amino acids and nucleotides, are moved into cells by facilitated diffusion. Despite the assistance of the carrier protein, this movement occurs through diffusion down a concentration gradient and is considered passive transport, since no energy is expended by the cell. Movement against a concentration gradient, however, costs the cell energy in the form of ATP. This process is called active transport. A carrier protein, such as the sodium-potassium exchange pump, uses energy to move ions across the cell membrane against their concentration gradient. In this example, ATP's energy is harnessed to move sodium ion out of the cell and potassium ions into the cytoplasm. This is the pump that maintains the differences in sodium and potassium ion concentrations between intra- and extracellular fluid.
What does a carrier protein do? What is facilitated diffusion? Does facilitated diffusion require ATP? What is active transport?
This concludes our section on the cell membrane. Next time you look at a doorway, think of the proteins that continuously transport molecules and ions into and out of your cells. Welcome to the Prentice Hall Video Tutor section on protein-to-protein interaction in muscle contraction. The activity of skeletal muscle tissue allows us to move our bodies. Without functioning muscle tissue, we would be unable to sit, stand, walk, speak, or participate in other usual daily activities. How do muscles move our joints? What happens in muscle tissue when our nerves signal it to contract? To begin to answer questions like these, we will look at skeletal muscle in 4 ways. We will describe the anatomical organization of skeletal muscle tissue. Then we'll examine the structure of a sarcomere, the repeating functional unit in myofibrils. Next, we will look at the link between action potential and muscle contraction, called excitation-contraction coupling. Finally, we will explain what happens during a contraction cycle. Skeletal muscles are surrounded by a layer of collagen fibers, the epimysium, which separates the muscle from neighboring bones and other tissues. Fascicles of the muscle are surrounded by another membrane, the perimysium, through which pass blood vessels which transport nutrients and waste products to and from the muscle, and nerves that provide the signal for contraction. Each fascicle contains numerous long, multinucleated muscle cells called fibers, and each muscle fiber contains numerous long cylindrical structures called myofibrils. Adjacent skeletal muscle fibers within the fascicles are bound together by delicate endomysium. An individual muscle fiber is surrounded by a specialized cell membrane called the sarcolemma. Invaginations of the sarcolemma form transverse or T-tubules that penetrate into the cell and encircle every myofibril. Also surrounding the myofibrils are the membranes of the sarcoplasmic reticulum, shown here in blue. The sarcoplasmic reticulum is an internal membrane network similar to the endoplasmic reticulum of other cells. Near the T-tubules, the sarcoplasmic reticulum expands into blind end chambers called terminal cisternae. The association of a T-tubule with 2 terminal cisternae is called a triad. Before going ahead, let's take a moment to review what we've just learned about the organization of muscle tissue.
What is a fascicle? What is the epimysium? What is the sarcolemma? Terminal cisternae are part of what structure?
Welcome back. We'll now take a close look at the structure of the sarcomere. Remember that each muscle fiber contains hundreds of cylindrical structures called myofibrils. The ends of each myofibril are attached to the sarcolemma of the muscle fiber. Thus, when myofibrils contract, muscle fibers shorten. A myofibril consists of a linear chain of sarcomeres. Sarcomeres are the smallest functional unit of a muscle fiber. They are composed of precisely organized proteins referred to as thick filaments and thin filaments. When these filaments slide past each other, the sarcomere contracts. Z-lines mark the boundaries between adjacent sarcomeres. Within a sarcomere, thick filaments are found in the A band. Thin filaments extend through the I band and into the adjacent zones of thick and thin filament overlap. The thick and thin filaments must slide past one another in order to make the sarcomere contract. Let's look more closely now at these filaments to see how the sliding is accomplished. Thick filaments are made of the protein myosin. Thin filaments contain the proteins actin, tropomyosin, and troponin. G-actin, here shown as red spheres, is globular actin. Each G-actin molecule has an active site that combines to the myosin of the thick filament. G-actin is strung together to make a twisted strand called F-actin. Tropomyosin molecules are positioned so that each covers 7 active sites along an F-actin strand. The position of tropomyosin in a resting muscle fiber prevents interactions between proteins of the thick and thin filaments. Midway along the length of each tropomyosin molecule is a molecule of troponin. Troponin has 3 globular subunits. One subunit attaches the troponin to tropomyosin. A second subunit binds to G-actin and holds the troponin-tropomyosin complex in position on the actin filament. The third subunit binds calcium ions. When the intracellular calcium level is low, the calcium binding site is empty. Tropomyosin molecules cover the myosin binding site on actin, and the muscle is at rest. When the intracellular calcium level rises, the calcium ions bind to troponin, causing a change in the position of the troponin-tropomyosin complex so that the active sites along the F-actin strand are exposed. Thick filaments are made of bundles of myosin molecules. Each myosin molecule has a double-stranded tail and 2 globular heads. In a thick filament, the myosin molecules line up so that the tails point toward the center of the sarcomere and the heads project outward toward the thin filament. The heads are arranged in spirals along the length of the thick filament. The myosin heads use energy from ATP hydrolysis to cock themselves into position to bind with actin. Knowing these structures of the thick and thin filaments will be important as we see how sarcomeres contract. Take a moment now for a quick concept check.
What proteins are found in a thin filament? The thick filament is made of what protein? Which molecule in the thin filament covers the sites that bind myosin? The binding of what ion causes a change in the positions of thin filament proteins?
Welcome back. Now we're going to look at the steps that lead from an action potential in the nerve to a muscle contraction. You might think of an action potential as the trigger for muscle fiber contraction. But how does this trigger work in muscle tissue? A motor neuron and the muscle fibers it activates are called a motor unit. In a motor unit, the neuron communicates with muscle fibers at neuromuscular junctions. When an action potential reaches the synaptic terminal at a neuromuscular junction, a chemical signal is released. The chemical diffuses to the sarcolemma of the muscle fiber and changes the permeability of the sarcolemma to sodium ions. The rush of sodium ions across the sarcolemma triggers the formation of an action potential that sweeps across the sarcolemmal surface. The action potential travels along the membrane of the transverse tubules. Its passage triggers the release of calcium ions from the terminal cisternae. The released calcium ions are bound by troponin. When this occurs, the troponin-tropomyosin complex changes shape. Now the newly exposed active sites of the thin filament can interact with the myosin heads that project outward from the thick filaments. The myosin heads bind to the exposed active sites, forming cross bridges that link the thick and thin filaments. We'll look more closely at how the cross bridges form and cause the filaments to slide. But first, let's have another concept check.
What is a motor unit? What is the name of the junction between a muscle cell and a neuron? Which sarcomere protein binds calcium ions? What organelle releases the calcium?
Welcome back. Let's see what happens now that the myosin heads have bound to the actin filaments. The cross bridges that form between thick and thin filaments must go through cycles of attachment and detachment so that the filaments smoothly slide past one another. Once binding occurs, the myosin head pivots, pulling the thin filament toward the center of the sarcomere. After pivoting, the myosin head binds a new ATP molecule, breaking the link between the thin and thick filaments. The ATP is hydrolyzed and the myosin head is cocked back into position to form new cross bridges with actin and begin another contraction cycle. The cross bridge formation is similar to the work your hands would do in pulling up an anchor. Cross bridge formation can continue for as long as calcium ions and ATP are present. The repeated pivoting cycles slide the thin filaments toward the center of the sarcomere, shortening the sarcomere and contracting the muscle. Remember, all the sarcomeres shorten together so the ends in each myofibril move closer together. You might compare this to what you see when an accordion is compressed. The outfolds correspond to Z-lines of adjacent sarcomeres. Just like the outfolds in this compressed accordion, the Z-lines move closer together during the contraction. After the contraction, there follows some passive expansion that returns the muscle fiber toward its resting state.
What molecule provides the energy for muscle contraction? Do sarcomeres lengthen or shorten during contraction? During contraction, how does the amount of overlap between thick and thin filaments change?
As we have now seen, all of our activities that use skeletal muscles depend upon the amazing organization and function of the sarcomere. Welcome to the Neurophysiology section of the Prentice Hall Video Tutor Series. If you've ever been on a crowded committee where many people are trying to make their ideas heard at once, it may seem remarkable that any one person hears or makes sense of what anyone else is trying to communicate, let alone collate all that information and make a decision. If you think that's impressive, imagine a situation in the brain where a single neuron may communicate continuously with thousands of other neurons. How is information transferred from one neuron to another in the brain? To begin to answer this question, this video tutor segment will cover several aspects of neurophysiology. First, we'll start with a look at the structure of a neuron. Then we'll define transmembrane potential and examine how neurons maintain theirs. Next, we'll look at how the movement of ions across the cell membrane affects the transmembrane potential.
Finally, we'll examine an action potential. We'll see how it is propagated along the membrane of a single neuron and how a stimulus is transmitted to another neuron across the synapse.
Neurons are the specialized cells that control and monitor body activities and physiological functions. They sense changing conditions, process sensory input, and direct the body's responses. Neurons come in many different shapes and sizes, but for all of them, the cell body, also called the soma, contains the nucleus and most of the other organelles. Extending from the soma are branch projections called dendrites. Their job is to receive information from the extracellular environment, from other neurons, or from other specialized cells. A typical neuron also has a long process called an axon that carries information that will be relayed to another neuron or a different cell type.
The axon ends in fine extensions called teledendria, each of which has an expanded synaptic terminal at its tip. A synaptic knob, shown here, is one type of synaptic terminal.
Inside each synaptic knob are synaptic vesicles containing chemical neurotransmitters that, when released from the synaptic knob, affect the transmembrane potential of another cell. How well do you know the structure of neurons? Take a few moments now to assess your understanding by answering 4 concept check questions.
What is another name for the cell body of a neuron? What cytoplasmic projection carries information toward another cell? Which cytoplasmic projections of a neuron receive information from other neurons? In what structure are neurotransmitter molecules stored in the synaptic knob?
Let's move on now for a look at transmembrane potentials. Intracellular and extracellular fluids have different ion compositions. Extracellular fluid has relatively high concentrations of sodium ions, which are positively charged, and chlorine ions, which are negatively charged. In contrast, an intracellular fluid, the most abundant positively charged ion is potassium, and most of the negative charges are carried by proteins. The negatively charged proteins cannot pass through the cell membrane, so we can think of these negative charges as trapped inside the cell. Both sodium and potassium ions, however, can pass through leak channels in response to the passive force of gradients. There are 2 gradients that exist across the membrane. A chemical gradient that causes the ions to diffuse to areas of lower concentration, and an electrical gradient, which causes ions of like charge to move away from each other, and ions of opposite charge to move towards each other. Let's first look at how these forces affect the movement of potassium. Potassium ions respond to both electrical and chemical forces. Although the electrical force opposes movement out of the cell to some extent, The chemical force is strong enough that potassium ions move down their concentration gradient and diffuse out of the cell through leak channels in the membrane. When these positive charges leave the cell, the interior of the cell membrane is left with an excess of negative charges. The charges carried by the intracellular proteins. Thus, the inside of the cell membrane is negatively charged compared with the outside. Sodium ions slowly leak into the cell. They follow both chemical and electrical gradients and enter the cell. The difference in positive and negative ion distribution across the cell membrane creates a polarization that can be measured as a transmembrane potential. The transmembrane potential of a resting cell is about -70 millivolts. Any shift in the transmembrane potential away from resting levels towards 0 millivolts or above is called depolarization. At the normal resting membrane potential, 3 sodium ions diffuse into the cytoplasm for every 2 potassium ions that diffuse out of the cell. The sodium-potassium exchange pump counteracts these movements. For each molecule of ATP expended by the cell, this exchange pump ejects 3 sodium ions and reclaims 2 potassium ions. In this way, the sodium-potassium exchange pump maintains the transmembrane potential and stabilizes the ion composition of the intracellular fluid. Let's stop here for a quick concept check.
At rest, is the interior of a neuron negatively or positively charged relative to the exterior? What is depolarization? What 2 forces contribute to ion movement across the membrane? What ion is brought into the cell by the sodium-potassium exchange pump?
Let's now take a look at how depolarization leads to an action potential. If the trigger of a gun is pulled just a small amount, the gun will not fire. For the gun to fire, the trigger must be pulled past a certain point. Similarly, the cell membrane of a neuron can depolarize a little without anything happening. However, once the neuron depolarizes past a level known as the threshold, an action potential is generated. Action potentials are depolarizations that start at the initial segment of an axon and are propagated toward the synaptic terminals. They are called all or none events, because either an action potential is triggered or it is not. There is no in-between state. Prior to an action potential, the neuron is at rest with a membrane potential of -70 millivolts. Now, if a stimulus depolarizes the cell to threshold, which is about -60 to -55 millivolts, voltage-regulated sodium channels in the membrane open. The sodium ions that rush across the cell membrane change the transmembrane potential rapidly from threshold towards 0 millivolts, where positive and negative charges are in balance. It then continues toward a positive potential, where positive charges outnumber negative charges on the inside of the cell membrane. When the transmembrane potential reaches positive 30 millivolts, the sodium channels close. The voltage-regulated potassium channels, now fully open, then allow potassium ions to flow out of the cell, a movement driven by both chemical and electrical gradients. As these positively charged potassium ions leave the cell, the transmembrane potential returns toward resting levels. The potassium channels begin to close when the resting potential is reached and finish closing shortly thereafter. During the absolute refractory period, the membrane cannot generate an additional action potential no matter how strong the stimulus is. This occurs while the voltage-regulated sodium channels are open and when they are closed and inactivated. The absolute refractory period typically extends until a transmembrane potential repolarizes to near threshold levels. During the relative refractory period, which occurs while the voltage-regulated potassium channels are open, another action potential can be generated, but only by larger than normal stimuli. Let's take a moment here for another concept check.
What is meant by all or none event? What happens to sodium channels when the transmembrane potential reaches positive 30 millivolts? What is absolute refractory period?
Now that we've seen how an action potential is generated across the neuron membrane, let's see how that action potential is transferred along the axon and eventually to other cells. An action potential can travel in one of two ways, either as a series of many small steps or in a series of a few leaps. When an action potential in an axon spreads to a neighboring region of its membrane by a series of small steps, the process is called continuous propagation. When it propagates by jumping from one site to another along the axon, the process is called saltatory propagation. Saltatory propagation occurs along axons that have myelin sheaths. In the peripheral nervous system, these myelin sheaths are formed by Schwann cells. The myelin acts as an electrical insulator, allowing ions to move across the cell membrane only at the gaps or nodes between adjacent Schwann cells. Therefore, action potentials rapidly travel from node to node. Once the action potential has traveled down the axon, its message must be passed on to the next cell. A neuron relays information to another neuron at a synapse. The neuron transmitting the information is called the presynaptic neuron, and the neuron receiving the information is the postsynaptic neuron. Let's look at what happens at a cholinergic synapse, one of the most common types of synapses. When an action potential arrives at the synaptic knob of the presynaptic neuron, voltage-regulated calcium gates open. Calcium ions enter and bind to synaptic vesicles. This leads to exocytosis, which releases the neurotransmitter acetylcholine from the synaptic vesicles into the synaptic cleft. Acetylcholine molecules then diffuse across the synaptic cleft and bind to ACH receptors in the membrane of the postsynaptic neuron. The binding opens ion channels and the membrane depolarizes. If this depolarization brings the initial segment of the postsynaptic neuron to threshold, it will result in an action potential. The depolarization of the postsynaptic membrane is short-lived because acetylcholinesterase rapidly breaks down the ACH in the synaptic cleft.
What does threshold potential mean? What is a synapse? What type of ion channels in the initial segment open when threshold is reached? What happens during repolarization?
Now, the next time you communicate with your friends, think of how all the neurons in your brain communicate with each other.